You all have decided to take a group picture or something?
Ma'am,
Yes,
this
ma'am.
t-shirt is given from the academy.
Academy.
So we were recording a video.
Oh, okay. So, didn't they give you a blue one? I thought it was a blue color one.
No, no, this time it
It
is
is
red.
nice. Yeah. So now, when is your last class? Not with me, but in general.
10th. Huh? No.
Next week, next Friday. Next Friday, you would have all been,
Next Friday is
Yes,
our main
Fridays are main
exam.
exam.
yeah,
Means 9th, ma'am. 9th is the last.
9th is the last day. So by next Thursday, you're all done with the SMS. So now you are going to train us with all your knowledge. Okay, so the structure of the course is such that it's taken you through the entire life cycle of SMF. I don't think even I studied all about what you are studying right now in my career. I mean, because you're getting exposure to the process equipment, the design of it, the various aspects which Viresh and the rest of the team has taken. We never went through that. We just heard from these specialists, and that's about it. But now you understand it better. So, what is the industry looking at you graduating from Biocon Academy or from a skill enhancement course versus what is it looking at when they're looking at an ordinary UG or a PG student? There is a difference. Like if I were to interview you coming in from Biocon Academy, that means I expect you to be more confident in correlating the various concepts. Like, for example, if I were to ask you, how would you handle a batch failure? What would you actually do when a batch fails? Then you should be able to pick up the relevant QMS elements, and then how it is handled, where will you go, and what are the likely issues that can happen during batch failure, especially related to a process equipment. These are the things that we expect you to speak on versus a UG or a PG who may just know the basic concepts of biotechnology or life sciences. They may not know as much. You yourself know, are aware that what you went through, the entire process, your knowledge has increased many folds versus what you were when you entered the course, right? So that's how you would have been if you were a fresher and I were interviewing you then. It would be mainly about, do you know quality, do you know GMP, those kind of typical question. But here we are looking at questions which are over here. Okay, so that means the skill has enhanced, and very, very important, they're looking at you to provide solutions. Please always remember, try to be a solution provider. When you have the thought process of being a solution provider, that means you will not stick to one specific domain. You will go across the various domain. What do I mean by domain? That means when you are giving your solutions, you're not only thinking of quality, you're also looking at the way the process equipment would have behaved or the strain would have behaved, the strain maintenance aspect, whether any issue is there with the strain, etc. So you're going to look at it from a bigger picture. So please keep that in mind. They're looking at you as a solution provider. You could say like an internal consultant. No? Okay. Deepika, Lasya, and Aditya having discussion. That's fine. So my main request is relook at all your slide decks for your sessions before your placements, and just go at it. And again, when you get a profile, when you get a job, please be in that profile for at least 2 years, because you feel you know everything by the end of 6 months. That is the attitude. Okay. And then you need it. See, 6 months is when you're actually knowing your job. The next couple of months, you're going to understand how your job is impacting the rest of the activities or how that is impacting you. Even I had the same attitude that I knew and I could head the lab and all that stuff that happens with confidence. Okay, there's nothing wrong with it, but tone it down, keep it even, be stable, be steady. Okay, try to navigate it. And again, when you are hired, of course, you will go through, I'm sure they'll teach you about interviewing questions, etc. Soft skill. If you are hired, couple of things is that you will be on probation for a couple of months because you need to get trained with their procedures. You are already trained with SMF. You know your, you know, concepts, but you need to undergo training in that particular area of work and those SOPs. That's the requirement of GMP or GLP, GCP. That's the requirement of the industry, so to speak. So please stay steady. Don't get influenced by people's comments. I've heard some of them saying, I've got calls later on from some students. I'm not telling you which batch, which college, which, whether it's Biocon Academy, nothing. Just to let you know that they have, people who are in the industry, they have told these students when they've gone that, what are you doing over here? With your education, you should not be in this role. You should be doing something else. Don't get carried away by their comment because they panicked and they called me and they said, am I in the right profile? They're saying that I should be in a much higher profile. Am I in the right profile? Ma'am, then I should probably, I need your help to get a different job. I said, no, you are in the right profile. Okay, your designation may be slightly different, your pay may be different. Don't reveal, of course, designation is something everybody gets to know. Don't reveal your pay, your offer letter. Don't go around telling your own peers over there who's sitting next to you. Don't reveal it. It's a very private information. It's as private as your, your medical checkup results. You cannot reveal it. And as per the company organization policy, you cannot reveal your, what do you call, pay. You cannot reveal your bonus. You cannot reveal a performance bonus. So please be careful because that can be against you. So please take care of your behavior. Please take care of your soft skills. Okay, please take care. Okay, so with that, we move on to the last two phases. That is, how do you store a product? So how do you store? Now that you have understood how the molecule behaves or the product behaves, how do you think it should be stored? There are two different things I want you to keep in mind. One is drug substance. The other is the drug product, DS and DP. In that, again, one is a chemically synthesized molecule. The other is a biologically synthesized molecule, right? Two
Yes, ma'am.
different aspects. So I am going to give you a small assignment again, like yesterday. And it's going to be interesting for you. Is that how would you, from the time of the strain, so one team, I want you to split into two teams. So today's session is all about storage conditions for drug substance and finished products, that is the final product. I want you to split into two groups. Okay, and I want one team to take up a chemically synthesized molecule. And I want the other team to take up a biologically synthesized molecule. Okay. And when it comes to chemically synthesized molecule, I want you to let me know how you are going to handle the storage of the product throughout its life cycle. Because they can ask you this during interviews. How would you store it? From the time, again, the raw material, the, what do you call, intermediates or in-process materials, the finished product of the API, then you're going to formulate that product. And again, it's going to be filled, say, in a vial, or it's going to be converted into a tablet. So what would be the storage conditions? How would you sort of identify what should be the storage conditions? It comes in the development. You and I know that it gets identified during the development of the molecule. Same thing, that's for chemistry. Now for biology, the product, from the time you receive the strain, how you're going to do the scale up, how you're going to manage it, about the storage throughout. From the strain, from the R&D, it moves to QA, rather QC, and then from QC, it moves to the manufacturing area. Then the scale up happens over a period of time. That means, say, 500 ml, 1 liter shake flask, then it goes into a big time scale up. And then that's upstream. Then in downstream, you will have something. Okay. Filtrations. After filtrations, then it goes through either tray dryer or it goes through solvent extraction, whatever. I'll leave it to you to identify whichever you feel like speaking about. And then you're going to convert it into a drug product. That is, the biological material is going to get converted into a drug product. You can take one example. And then how are you going to store it? Okay. So do you want me to go through the slide deck now or slide deck later? Today's sessions.
We'll do later, ma'am.
Okay. So you want to take this up. So it's now 9:15. How long do you think a time you need? Because 11:30 to 12:30, I need to switch over to the other subject. Yeah. So what time do you think? 9:15 now. Do you think 11 is good enough?
Yes, ma'am.
What do you say? Okay.
Okay.
I would say 10:45,
Okay.
and then we'll come back at 10:45. Okay.
Yes, ma'am.
Okay, ma'am.
See you.
Use precautions that should be taken in relation to the effect of atmosphere, moisture, heat, and light are indicated. During storage of pharmaceutical product is one of the fundamental concerns in patient's care. Then here first we got the general storage condition for small molecules. This we get from our SME in Syngene, and he told us that generally for small molecules, the raw materials will be stored at 2 to 8 degrees Celsius, not more than 25 degrees Celsius. And in process, while manufacturing, the temperature should be kept in a range of 5 to 10 degrees Celsius, and fill finish 2 to 8 degrees Celsius. And we took ciprofloxacin as an example. In that raw materials, ciprofloxacin, hydrochloride, and dexamethasone otic suspension should be stored at 15 to 30 degrees Celsius and protected from light. And freezing should be avoided. The raw materials freezing should be avoided. Ciprofloxacin hydrochloride tablet should be stored in tight containers at a temperature of less than 30 degrees Celsius. This is a temperature storage condition for ciprofloxacin as raw materials. In process, its temperature condition is 90 to 130 degrees Celsius. And as a finished product, this drug is available as tablets, IV infusions, and microcapsules. So the storage condition for tablet is 20 to 25 degrees Celsius. As microcapsules and diluent, its storage condition is 25. As infusions, its storage condition is 20 to 25. Also in refrigerator, we can keep it in a temperature of 2 to 8 degrees Celsius, but don't freeze it. That's all from team 1, ma'am.
Can you show me the material? Not material, this example. Can you come down? One minute, just hold it. Just hold it. When you're talking of raw materials, you're talking for the formulation. You're not talking about the API, right?
Ma'am, when I asked the SME, he said that the API can be stored in the raw, what are the Celsius, what is the degree Celsius mentioned in the raw materials? It can be stored likewise, he said,
No,
ma'am.
no, sorry, let me rephrase this. What I meant was, you're talking of raw material, that is the API, and then you're talking of the finished product also,
Yes, ma'am.
right? At the same time, in both in raw materials. Because suddenly in the beginning, first line, you're saying ciprofloxacin, hydrochloride, etc., stored at 15 to 30 and protected from light. Then in the next line, you're saying this hydrochloride tablet should be stored in tight containers. So is it then you say suspension in the first line and then you're talking of tablets. So
That is
what
a
is it?
raw materials. Ciprofloxacin hydrochloride is a raw material for the formulation of the drug ciprofloxacin.
Okay.
Mm.
And because if you look at it, Irene, in the first line, you're talking of the hydrochloride and suspension together.
Yes,
So are these two
correct.
both used as raw materials?
1 minute, ma'am. Let me check
Yeah, yeah.
that.
Yeah.
Ma'am, sorry, ma'am. Actually, it is a mistake. That is a finished product, ma'am. Actually, that is not a raw material. Dexamethasoniatic suspension is a drug, ma'am. That is not a raw material. We just got confused. Sorry, ma'am. And the drug name is ciprofloxacin, ma'am. The API compound in ciprofloxacin is ciprofloxacin. That is the chemical compound in that drug.
Ma'am, you are mute.
I'm so sorry. I don't know when it's gone into mute, but in the industry, now that you are aware of the requirements, you have the knowledge, may not be the entire knowledge because it's too vast. Not all of us know everything. Okay. So in these instances, you should be able to interpret the requirements fast. So tomorrow, what's going to happen in the team when you're working, they may ask you to do a lot of literature research, information gathering, okay, related to the product. You should be able to collate the information, but what value addition? See, collating information, even any UG PG can do. What value you're going to add is your expertise coming from the requirements. That is the regulatory requirements. So that will definitely add value. So here also, this will add value. Now, what was I inquiring here is from you. Would you agree that the raw material and the finished product should be at the same temperature, or does it vary from product to product? I mean, this particular product, what's your thought on this?
Ma'am, it will vary, ma'am.
Okay.
Raw material may be stored in a cold condition of 2 to 8,
Okay.
but in the finished product, it can be stored within 25 degrees Celsius.
Okay.
If it is a injectable product, means it will be stored in a freezer.
Cold. Yeah.
It is in a cold condition of 2 to 8 or 2 to 15, maybe.
Okay. Okay.
That's all, ma'am.
Okay. Thank you. So next team.
Good morning, ma'am. So we team two will talk about the storage of biological drugs.
Request you to speak louder, please. I can't hear you. Very, very feeble. Probably you
Oh,
can take another mic.
can you hear me, ma'am, now? Good morning, ma'am. So we team two gonna explain about the storage of the biological drug product. So here, the master cell bank is stored in the liquid nitrogen on its vapor phase, remains stable longer than those stored at -70 degrees Celsius. Whereas MCB, it is derived from the selected cell clone containing the expression construct. It is a starting point of a cell line development. Whereas a working cell bank, ideally, the storage temperature should be below -120 degrees Celsius, but typically it is best to go even lower below -150 degrees Celsius. Whereas WCB prepared from the MCB, it is used for manufacturing cycle. So here we took an example of insulin, which we will derive from E. coli strain. So first of all, from the bacterial strain, which we extracted from the cryopreservation, the temperature will be maintained at -80 degrees Celsius. So the bacteria, after the bacterial strain is taken from the cell bank, we will inoculate it, or we will culture it into a seed bioreactor, and the temperature will be 37 degrees Celsius, whereas we will maintain the pH of 5, and the stirring speed will be of 200 to 800 rpm, order to maintain the dissolved oxygen. So here the process result in a high cell mass, typically around 20 gram per liter. So then after the seed bioreactor, it will be then transferred to the higher bioreactor with a large quantity size. So the further information will be shared by Joshua.
Then further, after transferring into the large bioreactor, it will be multiplied into larger volume, made into a larger volume. Then after making into a larger volume, it is purified. Purification is done by the means of different techniques. Chromatography, filtration, and precipitation are used to purify. Other methods is used for purification. Then based on the we get the drug substance, and based on the stability studies, we can, according to the Q&A ICH, there are short-term storage, frozen storage, and intermediate storage. For the drug substance, can be for the short-term storage, can be stored at 2 to 8 for a shorter period, and whereas for frozen storage, it can be stored typically at commonly freezing temperature includes -20, -30, -40, and -70 degrees Celsius. And for intermediate storage, it can be stored at 30 degrees Celsius plus or minus 2 degrees Celsius with 65% of RH. Then for the drug product, insulin is stored at 2 to 8 degrees Celsius, whereas in special drug product, it can be stored at -80 to -60 degrees Celsius. For example, Ebola vaccines. Thank you, ma'am. That's all from team 2.
So what happens is, if you can go up, let's look at the working cell bank, master cell bank. So what do you think is the most critical aspect in the biologics manufacturing? What is it that's the crux of the biologics manufacturing, according to you?
Maintaining in its state, ma'am. Maintain its viability.
Even more than that, the main raw material.
The strain, ma'am.
The strain is a very, very critical, what do you call, raw material, though you could say a starting material. Why is it so critical? Any idea? Why should it be protected? So I don't know if I have shared with you my experience. I won't like to indicate the name of the client. When I was working in a company, this particular client had established a vertical over there, and they were planning to ship their not exactly master, but the strain from US to India. Okay, and what had happened was that before they shipped it, they wanted to ensure that the infrastructure was all okay. Why? Because I believe it's a very big company in America. I believe that company had shipped a strain many years ago, probably 15 years ago or whenever. They had shipped it to some other part of the world. And it so happened, I believe, when it was in transit, in transit, it stays in quarantine in the airport, because that's where the shipment happens, air shipment happens. So it was in transit in a particular warehouse, and somebody stole it, I believe. And that had a very high value, intellectual property value. That means it had a very high, very strong patent. That means it was so valuable. So because of that, they lost it. In the sense, losing your research, you can imagine. They lost a product, that is the strain. So after that, they decided, they relooked at their entire logistic network, everything, and they decided that they are going to audit their vendors, and they would audit us, because they're going to transfer the strain to us, and we would use it to scale up, and then we would be doing the manufacturing. So considering that, what they did was I faced for the first time in my life, in my career, an interesting audit known as asset protection audit. That means the strain was an asset and it had to be protected. And considering that the assets, it was, reviewed. So what happened was, it was an entirely, so my question to my senior was, my boss was, why should QA take it up? And then I was given the logic, which is actually right, is that strain is an asset. Strain is a very, very important, you could say, a raw material, starting material for the manufacturing. So it comes under GMP. So QA has to take it up. Because why did the question arise? Because we are used to getting audited for quality. And in this case, we were getting audited for the security. We were getting audited for the building. Of course, we were getting audited for supply chain and other things, but we were getting audited for security, building, then the fencing of the facility. Oh my God, it was a very, very thorough audit for about 8 to 10 hours. We had to prepare for it. In fact, they checked the camera. In the reception, they have a camera. So they wanted to know what is the pixel size of the camera. So very thorough audit, and all the teams cooperated well. They all were there. And in fact, we went and inspected the fence also, the compound, the biocon compound. So everything was inspected. And then finally, what happened? They went and inspected this particular company whose employee was with us then. And he was based in India with us, and we had given him an office area, and he had been given a lab, and there was a cold room, walk-in cold room, etc., etc. And of course, they had that company to whom he belonged to. He had a separate vault. He had a separate vault within the cold room. So you can imagine that what did that vault contain? The vault contained the strain. The vault contained the strain. And what do you say? This is how it happened. They checked whether the lab had a false ceiling. They checked whether the vault had a any false wall or roof. And then they also checked the reception area, whether those ceiling were false, etc., etc. About the window, about the opening of the window, everything, of the exhaust, everything we had checked. So this is how it worked. So the strain would be shipped to India through World Courier. It would be received. And at the receipt, typically the security or the customs people receive it, or the SEZ officials receive it. And this person had to go there personally, in person, go there and check the, check the package, verify it, and then the World Courier in their Jeep, they would come in their vehicle, they would drop him to his office, that is that area, that building, and then of course assist him in taking that particular carton up to his lab, and then of course into the cold room. And then of course, the vault would be opened only if you put the fingerprint. And then if he put the fingerprint is not enough, the other person, his counterpart from US, had to also put the fingerprint at the same time. So he had to coordinate with that person there. And then only the vault would open. So you can imagine the protection of the IP that they give. It's so important. Okay. Likewise, what happens is that when such kind of strains get received, after that activity, they would scale it up in their lab and hand it over to the QC and the QA. Basically, the QA would store it in the area in our area, and it would be under lock and key of the QA. They would have made, say, 10 vials. Okay, and they would have that particular account. Accountability is very important. Transfer of the strain from them to us, we had to note it down, and we had to, lock it. Nobody should have the access. It's only the responsible person. And then whenever the QC requested it, because they have to start the production, right? Based on this production schedule, and QC would come and request it and all that stuff. And then the vial would be handed over to QC. So this is how stringent it is. Okay, so this is how protective they are about their strain. So please be alert that these are very, very critical practices that the best-in-class industry follows. Okay, can you come down a little? Yeah, from the MCB. Okay, and then of course the WCB is done by the QC teams, and then of course it goes through the cycle. Okay, and then it's stored in the fermentation. Through the fermentation goes to the routine activity as per the temperature and relative humidity, etc., dissolved oxygen as per the BMR, and they process it. It goes into a reactor during the chromatography, column filtration, precipitation, everything. It will be as per the temperature condition. And in case they need to store in, for example, say filtration, that filter is occupied right now. So what do? They take drums, or it is stored in the particular reactor only. Again, depending on the plan they have, they will either store it in the cold room in the drum, or it will be stored in the chilled that particular reactor. No reaction will be occurring, but it still be over there. They do something known as hold time studies. These are very, very critically important, because you need to hold the material for some time, and that's where you need the information. Can you come down, please? Yeah, drug substance. This is all fine. Come down. Come down. Yeah, this is the last. Okay. And also when the material also gets distributed, the same temperature is maintained. Okay. So now good job, both teams. Fine tuning can be done, but yeah, I want you to understand this kind of assignments that you may be put through during the various activities in the industry. So I give you a break and we come back by 11:45. Is that okay? Yeah.
Okay, ma'am.
Thank you. We need to ensure that the drug is efficacious when it reaches the patients. So considering that these are very sensitive to temperature, light, they are appropriately handled. And we did discuss how it is done within the company. Let's understand a little more. And also in what do you call warehouses before it gets distributed, which is the next topic. These are stored in appropriate racks and shelves. And again, we need to ensure that the temperature is appropriately maintained and as per the prescribed condition. And again, based on the pharmacological action, that's how you notice that the pharmacist store the drug, or it could be stored alphabetically also. So now what happens is the company manufactures these drugs, and the company distributes these drugs into the various countries or to various pharmacies. Now, if you remember, the company will have its own licenses and regulatory approvals when they are manufacturing the product. Okay. Likewise, the warehouse or the pharmacist or the pharmacy where they store these drugs, they should have the license from the government. Okay. You and I cannot operate a pharmacy. Why? Because we are not pharmacists. Or if one of you is, then yes, you can. Pharmacists, again, have to be registered with the government and of course the state government also. They should display all those certificates. And that is for the pharmacist. For the pharmacy, again, they will have to have the licenses as per the DCGI requirements, and they will have the approval, and periodic inspection happens by the regulatory authority. This is for the regular routine drugs. Whereas if they are, scheduled H drugs, which are restricted drugs, which are as per restricted use, again, there are special permissions that are required to manufacture. There are special permissions and license that they need to have in the pharmacy also to store. You just cannot store it like a routine medicine. It has to be stored under controlled conditions. Okay. So again, the typical storage thumb rules. These are the thumb rules, remember. Ensure there is no contamination or deterioration or disfiguration of labels. Label disfiguration is a major issue. If in case you are unable to, or the label is not legible, or if it's torn, then you cannot consume that particular medicine. Please remember. Maintain the integrity and packaging. That means it's not tampered with. Prevent or reduce pilferage, theft or losses. Prevent infestation of pests and vermin. And again, it should not hinder the cleaning and have sufficient space for movement of stocks and handling. Okay. I'll probably have a discussion about this tampering thing, if not in this class, in the last class, or probably on Tuesday, about how tampering had affected one of the molecules or one of the medicines in the US. We'll discuss that. Again, damage you need to prevent to excessive stacking heights, and again, these are requirements as per the label. And the storage area will have a record temperature and it's monitored routinely. That means there will be probes across the area, and this is where they will monitor the temperature. Some of it is automatically done. We have something known as building management systems. These building management systems are also known as BMS, they call it as, but it's more of building management system that is all about temperature, power, all those requirements. Again, adequate temperature, lighting, cleanliness, humidity, cold storage facility, and very importantly, shelves. And again, to ensure that It's not going to get cross-contamination or deterioration. And well-closed container is a must. And protected from moisture means it should be stored in airtight containers. Again, protected from light. This is because some of them are photosensitive. And so again, you need to have put it in a container that will restrict that light pass. Again, temperature, refrigerator, what's it meant being in cold and what's it meant to be in room temperature. Again, there should be a good pest control activity. So pest control is a very, very important activity that takes place in the industry. And typically it is outsourced to a third party. And every room, every building, every area is under a pest control process. That means the vendor will have a schedule, and as per the schedule, they come and do the pest control. And the area in charge, like if you are the area in charge of your lab, then you are responsible to verify and sign up. And this pest control records is very critical because inspectors will look at it. Pest control is so important that they will review it. There are trend analysis that is done for pest control. And also if in case there are like some different species than what it is supposed to be there, then again, that has to be identified and appropriately there should be an action to minimize it or eliminate it. Again, the floor of the warehouse should be made of hard floor and must be in good state of repair and appearance all time. That means impermeable walls and floor. That is the thought process. That means if you were to see in the facility, you will notice that the wall will have a special coating of epoxy, sort of shiny kind of coating. But however, even the floor of the warehouse or wherever you're going to use should be in good condition. That means they should not be pitting on the, that means there should not be cracks on the floor or pits in the floor, because that can lead to contamination, microbe growth, and pests coming in and all that. So it has to be in good state of repair. And again, I don't know if you have noticed the wall junction. It's like, for example, the junction of the wall. Okay, this particular junction, if you notice, will be having coving. That is known as coving of the wall. Why? Because the insects should not penetrate the wall. The walls should not be porous. Just like how the reactors should not be porous. The reactors should be non-reactive in terms of, material that is put over there. So it has to be smooth. That's what they say for reactors. In case of here premises, the walls have to be smooth. Again, they add in they paint it with the epoxy paint. And again, you have the coving of the, the junction between the wall and the floor. And very importantly, the material, if you notice in the warehouse next time, it is kept away from the wall and at a certain height. The pallets are kept at a certain distance from the floor and the wall. Earlier, I'm talking long back, they used to use wooden pallets. But you and I know wood does have some kind of, insects in it. Again, you'll have to treat it and all that stuff. And that's why instead of that, we have got very good HDPE plastic, stacks. Okay. So the area used for storage of IV fluid should have adequate space and prevent exposure to direct sunlight. Again, each and every dosage form will have its own requirements. The warehouse area typically has 3 areas. One is when I'm talking of the warehouse of the raw material, for example, that will have 3 areas. Okay, one is receipt of the material. Okay, and the other one is quarantine and receipt of the material and quarantine. That's the area where the material is received, goods are received, it's verified, and label, everything is fine, they weigh it, they de-dust it, everything they do, and then they say, okay, now we can move this into the quarantine area. Quarantine area, that's where they verify all the requirements, the purchase order, invoice, everything, and they will put in a sticker, label. That is talking about the internal numbering batch, raw material numbering, etc. Then you will have the QC team coming there to sample the raw material. So they take square root of n+1. If it's 10 grams, then all the 10 grams. If it's about 10, then of course square root of n+1. Over there, what they will do is they will, stick that particular, there is a sampled sticker. They put that label on those quarantine sticker for the drums, whichever they have sampled. Okay, so if there are 20 drums, that means not all 20 drums are sampled, but square root of n+1 is sampled. So whichever drum you have sampled, you're going to put a label on it saying it is sampled. Then of course, once it's approved, then the label, on that label itself, they put in the approved label, and that moves into the approved area. In case they reject it, then it moves into the rejected area. So the rejected area is under lock and key, and typically it is under the control of QA. Okay, of course, expired goods and all that, they will discuss with the management, they will discuss with the vendor and all that, and they will take a call. Okay. Pest control I already said. So there are different type of pest controls. You have termite, rodents, then we have snake bite kits, and yeah. So, and of course, rat. Again, about shelving, it should be made of steel or treated wood. Again, you can't be having any rustic wood because you will have termites in it. It should be strong and it should be placed in alphabetical order, and empty space should demarcate one drug or dosage from another. Most recently received drugs are placed behind the old stock. That means first expiry, first out. FEFO, please note that down. This is known as FEFO or FIFO also they say, but if in FIFO, first in, first out, what happens is there could be some material which expires faster than the next lot coming in. So that's the reason why FEFO is better. Put the lids properly, ensure everything is maintained well. I think, yeah, here it's given. The longer expiry dates should be placed behind those with the shorter expiry dates. Again, clean after each use of tablet counters in the dispensary and place within easy reach on the table. Avoid dispensing wrong drugs by arranging drugs on the table in alphabetical order. Always close drug containers from which drugs are not being dispensed to prevent spill or dispensing the wrong drug. Drug expiry, the time where the given product stored under reasonable condition is expected to remain stable. And essential drugs that satisfy the health needs of the majority of the population. So in terms of expiry dates, medications must be administered and products and equipment must not be used beyond their expiry dates. All medical equipment, dressing, solution used during invasive procedure must be sterile. Single-use devices are meant for single use only and must not be reused. There is a bad habit, many people do that. All drugs should be maintained separate from non-medications in a locked cabinet, which is sufficiently secure to deny access to unauthorized people. It should be available only to authorized persons who are assigned medication-related responsibilities. Medications that are used for external use only, separate from medications intended for internal use. So these need to be stored separately. Store look-alike and sound-alike drugs together, separately. Maintain the temperature. When refrigeration is necessary, use a medication-only refrigerator. That means do not keep any food items within it. Verify and document the temperature. All multiple dose injectable medication should be initiated and have the date of first entry recorded on the label. Rotate medication stock monthly using IFO. Two licensed personnel count every shift. Witness all the discards. Record on control substances sheet all administrations and wastes. All details must be completed in doctor's own handwriting, like name of the drug, dose of the drug, number of dose, signature of prescribing doctor. They must be kept in a locked cabinet. It should be in possession of an authorized person. Students no way are responsible for it. It must be kept locked in a locked cabinet. It must be in possession of the authorized people. It must be a trained nurse or midwife. The students should not be responsible for controlled drug cupboard keys. Everything needs to be recorded. How much you issued, how much is there. Everything needs to be documented. Any questions? So Irene, I have received your request, and my colleague has shared it with me. She has shared it with me. I will send it to you by end of the day today.
Thank you, ma'am.
Yeah. We move on to the next penultimate class. So we will be going into distribution requirements for distribution requirements for drug substance and finished product. I'll quickly go through it and I'll give you your assignment. On Monday, we can discuss it. Just briefly, this is, yeah, will not take
Ma'am.
time.
Ma'am, do you have sessions on Tuesday evening?
Tuesday morning, no?
Okay, so I got a calendar from Nitya ma'am. In that it is mentioned that Tuesday evening environmental monitoring by LAKSHMI ACHUTA is given.
What is that Tuesday evening?
Environmental monitoring.
No, that's already done. That I did last week, remember?
Yeah, that is what I'm asking. Why they are mentioned
I
it
think they
again?
mentioned it by mistake, I think, yeah.
Then, ma'am, are you taking power management for us?
Mhm.
Okay.
Somebody else, I think they have assigned.
Okay.
I wanted to ask you about, I think Tuesday morning is my session at 9.
Okay, ma'am. So it will be the last session, right?
Yes.
Okay.
So today I will take up the next one, distribution requirements for drug substance and product. Quickly, let me just go through it. And it's something similar to what we just discussed. Again, good distribution practice, GDP it's called. Earlier, GDP used to be good documentation practice. Now it's called as GDP, good documentation practice. So what does GDP say? Minimum standards that the distributor must have to ensure for quality and integrity of the medicine. And they agreed to abiding by the requirement. And it will be carried out as per the principles of GDP and ensure that there is no mix-up, adulteration, contamination, cross-contamination, and spurious drugs. Again, to maintain the quality and identity of the product. throughout the life cycle. It is to apply to individuals and organizations involved in the storage and distribution of pharmaceutical products. It also applies to sourcing, storage, transportation of API and other ingredients used in the production of the medicines. So, all parties involved in the trade and distribution of pharma are covered by this, including the bulk finished good, wholesaler, supplier, distributor, government organizations, quite a list of people. So the guidelines across the globe are somewhat similar. They outline the practices that needs to be done. So some of them are related to personal, quality system, premises, documentation, traceability, complaint return, transportation. Again, as per the law, national legislation is a must. This is what I was telling you. There should be a license. They should have a registered business if you look at that distributor. And again, they should have the authorization to import or export. Pharmaceutical products should only be supplied by distributor or their agents to individual or organizations who are legally permitted to receive. The subcontractor must have required authorization. Only registered or approved mail order pharma or their authorized company should be able to sell medicinal products online. Designated people are responsible for compliance. It's complicated. So you could say they have to be experienced and trained and qualified people. Training and maintenance of training records should be there. Again, hygiene, organization role should be very clearly defined. Code of conduct should be there. Quality policy should be there. Designated person to assure quality system. Attain a certificate of compliance with ISO quality standards. Ensure good storage practice. I discussed about it with you. Precaution policies should be there. Storage area for the various type of drugs should be there. First expiry, first out should be there. HVAC system should be available. Again, all documents should be appropriately written down. They're complete, approved, and authorized. Documents that are comprehensive with clear and specific language should be there. And record of defined amounts of time specified by the national law. Prepare procedures for traceability of products distributed to facilitate recall. Very, very important. Product recall. So what happens when the company realizes that there is an issue? Then they have to voluntarily recall that entire material into the system. Identify all the stakeholders involved. Develop the product coding systems. Again, record all complaints and recalls. I think we've already discussed this. Communicate about it, investigate it, write procedures related to it or write the report related to it. And ensure physical segregation and storage of return stock from the saleable stock. Separate warehouse only will be there. Vehicles must be dry and free from insects to prevent pilferage. Where special storage conditions need to be maintained, these should be provided. Individuals responsible should be informed about all conditions for storage and transportation. Technology such as GPS-enabled electronic tracking devices and engine kill buttons should be there. So here, company have gone way ahead. They not only have that, whenever they're shipping, especially sensitive material, they have something known as temp tail. That means they put this particular, it's a device which records the temperature and the what do you call, records it, and it produces a report. So in case you're shipping from India to say Europe or UK, then that if you put in the temp tail inside it, it'll record it. You have to start it, of course. It will record it throughout. And then when you return back and hand it over to them, it will tell you how much you have used. Okay. And what is the records of those temptail recording. And if there is any variation, then automatically we'll have to raise a deviation and take it forward. Self-inspection is something also known as an audit. It should be conducted and it has to be sort of recorded. And it has to be as per the schedule. So let's look at distribution chain, active API, manufacturer, distributor, pharmacy, and patient. Whereas in case of repackager and distributor, it goes through these activity. So it reduces distribution complaints, mistakes, achieve consistency, reduces weight. Okay. Again, the vehicles and the equipment used should be in such a manner that no issues are there, including, especially related to cleaning and safety. And it should be kept clean, free from accumulated waste, free from rodents, vermin, birds, and other pests. Sorry, it's written as pass. A written cleaning program should be available, negating the frequency of cleaning and methods to be used. So what happens in some cases, some companies, they repackage and relabel. So that's again, they need a permission from the government, and accordingly, they'll have to receive it, repack it, and relabel it. This is typically seen in case of APIs. Another threat that is happening is related to the counterfeit pharmaceutical products. Any such thing observed should be segregated immediately and recorded and of course investigated, etc. It should be clearly labeled to prevent further distribution or sale, informed immediately to the holder of the marketing authorization. A nation's port of entry for the processing of pharmaceutical product import should be restricted by suitable laws. Pharma products should be stored under suitable conditions as short time as possible. So if you look at EMA, that is the European Medicinal Agency, there are requirements related to contract activities. Any activity related to distribution of pharma product, which is delegated to another person or entity, should be performed according to the terms of the written contract, which is agreed. The whole distribution of medicinal product is performed according to the information on the outer pack. The wholesale distributor should also use all means available to minimize the risk. Of course, risk assessment is part of it. Now coming to brokers, they are a person involved in activities in relation to the sale or purchase of medicinal product, except for wholesaling distribution, because that does not include physical handling and consists of negotiating independently and on behalf of another legal or natural person. Any questions? So I have a, quick assignment, which you can, again, anyway, split into two groups. I want you to just, you have given me about the storage conditions during manufacturing. Now I want you to let us know about the storage condition after it is shipped out and it's getting distributed. We'll discuss that for about 10-15 minutes on Tuesday. Okay. Fine. I end my class for today. Okay. I will discuss a case study on Tuesday, and also we will discuss about this small molecules and large molecules, how we are going to distribute it in the requirement, as per the requirement. Okay. Thank you very much. Have a nice day and have a good weekend.
Thank you, ma'am.
Thank you, ma'am.
Thank
Thank
you,
you,
ma'am.
ma'am.
Thank you, ma'am.